right now, like a meter beneath your boots, the dirt is actually breathing.
It really is.
It's inhaling oxygen and feeding this microscopic, sticky city that is quietly doing something a billion-dollar water treatment plant actually struggles to do.
Yeah, and it sounds like science fiction, honestly. I mean, we are so conditioned to think that water purification requires, concrete facilities, chemical scrubbing, and just absolute visible control.
Right.
But the reality is that the ground itself is a highly efficient bioreactor.
Which is incredibly relevant to you if you, say, manage a local irrigation system, if you farm crops, or honestly, if you just turn on the tap every morning.
Exactly.
Because groundwater depletion is a massive looming crisis. I mean,
Yeah.
the wells are running dry, and traditional treatment plants just cannot scale up fast enough to replenish our aquifers.
They really can't.
So we are forced to look at natural filtration, basically shifting from precision engineering into a landscape that is, literally murky.
Right, so we are talking about managed aquifer recharge, specifically a method called soil aquifer treatment.
Okay.
It's basically nature's ultimate recycling program. You take treated wastewater, let it soak through the ground, and just allow the earth to clean it naturally before it hits the underground water table.
And that brings us to the mission of our deep dive today. We're looking at a 2026 paper from the journal Hydrology and Earth System Sciences.
Yeah.
The title is Mapping Water Content Dynamics in SAT Systems Using 3D Electrical Tomography.
Quite a mouthful.
It is. But the goal here is to uncover the specific soil recipe and the exact watering schedule you need to maximize this natural filtration without, accidentally clogging up the whole system and flooding the surface.
Right, and the premise of this study really shifts the standard regulatory focus, because usually authorities obsess over the chemical composition of the water before it enters the ground.
Yeah, exactly.
But this research team asked a totally different question. They asked, what is actually happening down in the dirt to process those chemicals? And how can we engineer the soil to do it better?
So let's start with the dirt itself, because I always assumed the ground acted like, a giant coffee filter or a physical sieve.
Most people do.
Right? Like you pour muddy water through sand, the sand catches the solid particles, and clear water drips out the bottom.
Sure.
But this paper focuses on the unsaturated zone, which is that layer of damp or dry soil sitting just above the water table. And it reveals that physical filtering is like only a tiny fraction of the story.
A very small fraction, yeah. The unsaturated zone is this incredibly dynamic environment because it contains all three states of matter at the same time.
Oh, wow.
Yeah, you have solid soil, liquid water, and atmospheric gas. So when you introduce the organic nutrients found in wastewater to the bacteria living down there, they don't just float around.
What do they do?
They actually alter their environment. They group together and secrete these sticky layers, which researchers measure as extracellular polymeric substances, or EPS.
Okay, so the bacteria are basically manufacturing a type of glue.
Exactly.
Why, though? Is it like a defense mechanism against the flow of the water?
It's pretty much a survival strategy. These biofilms anchor the bacteria to the grains of sand or soil.
Correct.
And as the wastewater trickles past, this sticky layer acts like a localized microreactor. It physically traps pollutants, pathogens, and specifically these stubborn lipophilic organic compounds.
Lipophilic meaning the toxic chemicals that just refuse to dissolve in water.
Right, exactly.
Okay, so they trap them, but then what? Did the pollutants just sit there indefinitely in the glue?
No, the bacteria actually consume them.
Wait, really?
Yeah. Through biodegradation, microorganisms catalyze oxidation reduction reactions. They literally use these organic compounds and pollutants as a food source.
That's amazing.
It is. The biofilm holds onto the water and the pollutants simultaneously, which creates the perfect microenvironment for the bacteria to just break down the toxins over time.
Wait, I do see a glaring paradox here, though.
Oh, what's that?
If these bacteria are constantly secreting biological glue to trap pollutants, aren't they sort of sealing their own coffin? Like at some point, that glue is going to fill all the empty space, turn the dirt into concrete, and the water just won't be able to drain at all, right?
Spot on. You have identified the primary bottleneck of soil aquifer treatment. It's called bioclogging.
Bioclogging.
Right. If the biofilm grows too thick, it completely fills the pores between the grains of dirt.
Yeah.
Infiltration stops, and the wastewater simply pools on the surface.
And the whole thing breaks down.
The entire system fails. So the real engineering challenge is finding a way to encourage enough bacterial growth to clean the water, but prevent them from, choking their own habitat.
Which brings us to the real-world experiment. To find this delicate balance, the researchers set up a pilot test at a wastewater treatment plant in Palamos, Spain.
Yep.
And they built two separate 3.5 square meter basins. System 1 was a traditional filter made purely of fine sand, which I assume is just the industry standard.
It is, yeah. Plain sand offers very predictable drainage. But for System 2, they designed what they termed a reactive barrier system.
You know, the phrase reactive barrier conjures up images of like synthetic lab materials or glowing membranes or something.
Right, something very sci-fi.
Yeah. But the actual recipe they used is surprisingly earthy. Like it's 50% sand, 30% organic matter, which is specifically a mix of wood chips and garden compost, 10% biochar, 8% zeolites, and 2% clay.
Yeah, it's essentially an engineered soil designed to optimize both chemical retention and biological growth.
I'm actually super curious about the organic component there.
Okay.
Does it matter what kind of wood the wood chips come from? Like, is oak better than pine for growing these biofilms?
The paper does not mention that.
Oh, gotcha. But it does detail the mechanical purpose of those wood chips, right? Like, they aren't just in there to slowly rot.
Exactly. They serve a dual purpose. First, the wood chips and compost provide an immediate, rich carbon source. It's literally food for the microbes.
Okay.
Second, and perhaps more importantly, they provide what we call secondary porosity.
By secondary porosity, do you mean the water isn't just flowing between the grains of sand, but it's actually hiding inside the physical structure of the barrier materials?
Consider the geometry of it. Plain sand has a porosity of about 38%, meaning 38% of the volume is empty space for water to flow through.
Right.
The reactive barrier, however, utilizes the irregular shapes of the wood chips, the microscopic honeycombs inside the biochar, and the zeolites.
Oh, I see.
So it acts like a complex sponge made of mismatched puzzle pieces. It holds a porosity of roughly 59%.
Wow, that is over 50% more open space than just plain sand.
Exactly, which means there's way more surface area for the bacteria to colonize without immediately blocking the main pathways for the water.
But did it actually translate to more bacterial growth? Because to measure this, the researchers had to take core samples of the dirt every week.
Yes, they did.
They put them in sterilized tubes, took them to the lab, and used a cation exchange resin to literally shake the dirt for an hour.
Like trying to extract it.
Yeah, trying to extract and weigh that sticky EPS glue without destroying the actual microbial cells themselves.
And the data showed a staggering difference, actually.
Really?
Yeah. In the plain sand system, they measured roughly 70 micrograms of biofilm material per gram of soil.
Okay.
In the reactive barrier, that number spiked to nearly 390 micrograms per gram.
Whoa, that's like over 5 times the amount of biofilm.
It is. The organic-rich barrier created a massively thriving microbial ecosystem, and it triggered this really powerful feedback loop.
How so?
Well, the barrier holds more moisture due to its high porosity, right, which allows the biofilm to grow.
Right.
But the biofilm itself is hydrophilic, meaning it aggressively holds on to water, which then further increases the...
Oh, so it just supports even more bacterial growth.
Precisely.
Okay, so the reactive barrier is clearly the superior habitat, but the bacteria are trapped a meter underground under a literal flood of wastewater.
Right.
How are they not suffocating? Because earlier, we established that the unsaturated zone requires gas. The bacteria need oxygen to catalyze the reactions that break down the pollutants.
And that introduces the operational side of the experiment. Having the optimal soil chemistry is kind of irrelevant if the physical delivery of the water is flawed.
Makes sense.
So the researchers tested 2 distinct watering strategies over several months. The first was a continuous operation.
Meaning just pouring the wastewater into the basin nonstop, 24 hours a day at a steady flow rate.
Exactly. And the second strategy was a pulsed operation. They poured water for 12 hours and then completely shut off the flow for 12 hours.
So to make sure the total daily volume of water remained exactly identical, they just ran the pumps twice as fast during the 12-hour on cycle.
Right. And this pulse strategy is designed to induce a phenomenon known as aquifer breathing.
Aquifer breathing. So pumping twice as much water for half the time, then letting it sit. Is the goal basically to let the water drain so the soil can physically inhale atmospheric air, like catching its breath?
Physics dictates that it pretty much has to.
Really?
Yeah, when the water flow stops during that resting phase, gravity continues to pull the existing water downward.
Right.
And as that heavy liquid vacates the upper pores of the soil, it creates a negative pressure zone, basically a vacuum.
Oh.
That vacuum forcefully sucks fresh atmospheric air down from the surface, deep into the dirt.
Delivering a completely fresh supply of oxygen directly to the biofilms?
Exactly. Without that oxygen, the system goes anaerobic. The entire microbial community shifts, and the degradation of those critical contaminants just slows to a complete halt.
And the results of the 2 watering schedules seem to prove this perfectly, because the pulsed flow caused an immediate massive spike in healthy biofilm growth.
It did.
I mean, the combination of deep oxygen penetration and the extra heat during that specific testing phase just sent the EPS production into overdrive.
Yeah, whereas the continuous flow demonstrated exactly what happens when you deprive the system of that breathing cycle.
Because the continuous run only lasted about 91 days before it completely failed.
Right.
A puddle formed on the surface, infiltration stopped, and the system choked. I would assume the biofilm just grew too thick because of the constant food supply.
Well, the bioclogging was definitely a factor, but the failure was actually a compounding cascade of physics and biology.
How do you mean?
Without a resting period, the fine organic particles suspended in the wastewater simply accumulated on the surface. There was never a dry period to allow the biofilm to die back, compress, or even crack open.
So it was just in a perpetual state of saturation.
Exactly. Furthermore, the continuous test ran right into the winter months. And as temperatures drop, the physical properties of water actually change. Cold water is more viscous than warm water.
Wait, it gets thicker?
At a molecular level, yes. It has a higher resistance to flow, meaning it moves significantly slower through those microscopic pores in the soil.
Oh, wow.
Add to this the fact that the surface grass planted on the basins was dormant in the winter, meaning no new root growth was actively breaking up the topsoil, and the surface simply sealed shut.
So how did the plain sand and the reactive barrier compare during these schedules?
Well, the plain sand behaved very predictably. During the 12-hour dry periods, it drained rapidly. The oxygen penetrated deeply, and the sand dried out.
Okay.
The reactive barrier, conversely, remained heavily saturated.
Because of the wood chips and the massive amounts of that sticky biofilm holding onto the moisture.
Right. It basically acts as a reservoir. Retaining that moisture is highly beneficial for keeping the bacteria alive and actively digesting pollutants during extended dry spells.
That makes sense.
However, it requires intense management. Because the reactive barrier naturally holds so much water, if the resting pulses are too short or the climate becomes too cold, that high moisture retention will push the system into total bioclogging much faster than plain sand.
Okay, but all of this complex oxygen sucking and biofilm growing is occurring a meter underground. If the researchers cannot physically dig up the soil without completely destroying their own experiment, how are they measuring the moisture retention? Like, how are they actually map where the water is going?
They utilize 3D electrical resistivity tomography, or RT.
Looking at the data visualizations in this paper, RT looks exactly like a medical MRI or an X-ray for the Earth.
That's a great comparison.
How does it actually see through the dirt, though?
By measuring electrical resistance, the researchers drove 6 plastic rods deep into the soil, down to the bottom of the unsaturated zone.
Okay.
And each rod was equipped with 12 electrodes. These electrodes emit small electrical currents and measure how difficult it is for the current to travel through the soil from one point to another.
I am trying to connect the dots here. Why does the electrical resistance tell us anything about the water itself? Is it because the wastewater is full of minerals?
Precisely. The treated wastewater pumped into the system contains dissolved salts and ions. It is highly conductive.
Ah, I see.
Therefore, if the ear rate detects a zone of low electrical resistance, it means the current is flowing effortlessly, indicating that a specific patch of soil is heavily saturated with wastewater.
And a zone of high resistance indicates dry soil.
Exactly.
So they essentially map the moisture by mapping the electricity. And the paper actually notes a super fascinating real-world variable that occurred during the scanning process.
Right, the storm.
Yeah. The pilot plant was drawing water from the local Palamos municipal sewer system,
which is a coastal town. And during severe windstorms, the ocean waves would actually breach the coastal defenses and flood seawater into the town's sewage network,
causing the wastewater entering the experiment to become suddenly aggressively salty.
Yeah, and the researchers turned this anomaly into a massive advantage. That spike in salinity drastically lowered the electrical resistance.
Wow.
So by watching that specific pulse of highly conductive water travel through the E-rate map, they could track the exact velocity and behavior of the water moving through the soil.
Which leads to what I think is the most surprising revelation in the paper. Because I always picture water sinking into the ground in this flat, uniform sheet, like a blanket slowly being lowered.
That's how we usually draw it in diagrams.
Right. But the ART scans prove that is completely false.
Absolutely false. The infiltration is entirely heterogeneous. The water forms what hydrologists call preferential flow paths, or fingers.
Like raindrops tracing erratic paths down a window pane, just finding the route of least resistance.
Exactly. Gravity, combined with microscopic variations in soil density, forces the water to channel into these vertical columns.
And you can see that on the scans.
The ERT maps visually confirm this. You see these dark, highly saturated zones snaking downward, leaving adjacent pockets of soil completely dry.
But the ERT didn't just track the water, right? It actually managed to track the bacteria.
It tracked the physical signature of the biofilm, yeah. During the pulsed operation, the researchers shut off the water flow and allowed the system to drain for 4 hours. Now, gravity dictates that the highly permeable sandy soil should empty out. Yet when they took a subsequent ART scan, the maps revealed isolated super wet spots that stubbornly refused to drain.
And those spots couldn't just be random puddles if the soil around them was porous enough to let the water fall.
Right. Based on the physics of the soil, the water should have vacated those pores. The only logical conclusion is that the ERT was detecting massive clusters of biofilms.
The glue.
Yeah, the EPS glue was holding onto the water, acting like a completely saturated sponge suspended in the dry soil.
This implies a wild physical feedback loop.
Doesn't it?
Yeah, the bacteria aren't just reacting to the water flow, they're actively dictating it. Like they colonize a preferential flow path, they secrete glue to trap moisture, and in doing so, they physically block that channel.
They literally alter the architecture of their own environment.
That's incredible.
It is. As they clog their initial pathway, the subsequent wave of wastewater hits that biological roadblock and is forced to channel in a completely new direction. The researchers actually noted that these saturated hotspots migrated over the course of the experiment. The microbial city is constantly shifting, chasing the nutrients in the flow.
It's just an entire shifting, breathing ecosystem operating right under our feet.
Truly.
So stepping back from all the ERT maps and the chemical analyses, what is the ultimate takeaway here? Like, why does a utility worker, a farmer, or even just a local resident need to understand the porosity of wood chips or the viscosity of winter water?
Because global water scarcity is forcing our hand. Our reliance on treating and reusing wastewater will only increase.
Right.
And this research demonstrates that achieving high-quality water purification does not strictly require massive capital investments in high-tech concrete filtration plants.
We can leverage the dirt.
We can engineer the biology of the soil by utilizing inexpensive, globally accessible materials like wood chips, compost, and biochar.
Just cheap, natural stuff.
Exactly. And pairing them with a scientifically timed, pulsed watering schedule, we can harness the bacteria. We can build natural reactors that efficiently digest toxic pollutants, provided we manage the breathing cycles to prevent bioclogging.
And aerate technology simply gives us the visual monitor to ensure the aquifer's lungs are functioning.
Right. It's a profound shift from brute force engineering to ecological engineering. Instead of just pumping water through a static filter, we are cultivating an environment that does the work for us.
It requires a pretty nuanced understanding of the physics and the biology, but the application is incredibly practical for communities facing immediate water shortages.
Highly practical.
Which leaves a totally fascinating, forward-looking question to consider.
Okay, let's hear it.
We know that the bacteria living inside a simple wood chip and compost barrier are doing the heavy lifting, secreting glue to capture and digest pollutants.
Right.
We also know that different types of bacteria thrive on different carbon sources.
Yes, they do.
So as this ecological engineering advances, could a local water utility eventually design completely custom soil recipes? Perhaps like a targeted industrial probiotic blend engineered specifically to hunt down and digest unique, hard-to-remove chemical spills plaguing their specific region.
Well, once we can map the subsurface flow with ER and we fully understand the biological requirements of the bacteria, customizing the ecosystem to target specific contaminants is definitely the logical frontier of this technology.
It just fundamentally changes how you look at a patch of damp ground. It isn't just mud and water. It is a highly sophisticated microscopic treatment facility, quietly inhaling oxygen and just waiting to be put to work.